So before we start submission, it's very important to understand how our metadata is structured and linked together. This model that I will present is actually the same model that's used for all types of nucleotide data across all species, which is why it's perhaps a little more complex than some other systems. But I think it makes a lot of sense in terms of how it's broken down into searchable objects. So this is the structure of the model. I'll go through them very quickly, but we will go into a bit more detail in a minute. And I'll go through them in the order that we should submit them. So first should be the study. This will define release and ownership and affiliation of the data, and it will really hold all of the other objects that you will submit a bit later. Next, you should submit samples. This is defining metadata about the sequenced biomaterial. So in our case, that will be the actual virus itself. Then put third and fourth with a star. You may submit sequences without raw read data, but if you are going to submit raw reads and sequences, we recommend you submit the raw reads first, and then later the sequences, which you can then link to the raw reads. So we'll go into a bit more detail about each of these now. As I mentioned, study really binds all the related objects together into a cohesive unit or project. When you submit, you will receive project accessions like PRJEB and ERP. A PRJEB is a bioproject accession, while ERP is an internal ENA accession, and I'll discuss this a little bit later too. If you're going to reference your data in a publication, we always recommend that you reference at a study level. That's because if you discover some issues with your data a little later, it's quite easy to kind of suppress some parts of it and re-upload the correct data, and it'll all kind of happen transparently inside the study. The example metadata that is held on a study is a title and description. What is the study about? What's the aim? Sometimes we have taxonomy at a study level. If everything in the study is on the same species, then it's good to add the taxonomy at that level. Affiliations, so submitters, authors, centre names, consortium, that sort of level of information can be added to the study. And of course release date. So you can have studies that are held privately, where you can define a list of collaborators who can see this private data, and when you have finished with your analysis and you're happy for the data to go public, we can then release it. So yeah, release date can define at what point it actually goes fully open to the public. Next we have samples. So as I mentioned, that's defining metadata on the sequenced biomaterial, which is the SARS-CoV-2 virus. In our case, you'll get accessions like SAME and ERS. SAME accession is a biosamples accession, and ERS is the ENA internal accession. And the type of metadata that we would define on this level is obviously taxonomy is essential to tell us what the actual sample is. Collection date and location at least country level are also quite essential, just to make this data useful, searchable. Host information is greatly encouraged, so things like the age, sex of the host, disease outcome, have they recovered, etc. and lots of other information too, like vaccination status, and there's quite a range of host information that can be added to these samples. Just to quickly mention a checklist, but I will come back to this a bit later. So of course this metadata model is the same that's used for all different species. So of course things like vaccination status is a good example. That doesn't apply if you've sequenced a, let's say, a penguin. So we have different checklists for different types of sample. The sample checklist for viral pathogens is ERC 000033, and I'll come back to this a little bit later. But there are different checklists for, let's say, metagenomes or kind of sewage samples, for example. They all have different sets of metadata that should be associated with those samples. Raw read data is a little unusual in that it comes in two objects. So the experiment really holds the information about how what kind of protocols were used for the sequencing experiment, and then the run object really just holds the data files themselves. So experiment accessions look like ERX star, and the type of metadata that you'd kind of expect on that level are instrument platform, what kind of sequencer did you use, what model did you use. We even take things like the library prep information, what primers were used, kind of construction protocols, that sort of kind of level of information. And yeah, as I mentioned, the run holds the actual files that come off the sequencer. We support all sorts of different file formats, but obviously BAM, CRAM, FASTQ are kind of the most common, and those accessions look like ERR. So yeah, as I mentioned earlier, we can accept sequence data alone without any raw reads, but if you do want to submit both, we recommend submitting the raw reads first and later the sequence data. That is because there is a metadata field on an analysis object called runref, which allows you to define the ERR accession that the sequence was built from. So analysis accessions look like ERZ. The metadata that you'd typically see on this type of object is the analysis type. Almost always, in the case of SARS-CoV-2, that is sequence assembly analysis type. The other types of kind of assembly-related metadata would be the method, the platform, even the software that was used. And a lot of those fields are free text, so it's quite acceptable to put as much or as little information as you'd like in there, but obviously we would always recommend giving as much information as possible. Depth of coverage and molecule type are some other examples. So you've probably noticed that in a couple of cases you could receive a few different accessions for what would appear to be the same object. Study or project receives ERP and PRJEB accessions. So the real difference here is that there's kind of internal accessions for objects within ENA or SRA in the case of the NCBI side, and then there's ones that are shared across all of INSDC. So that will include ENA, NCBI and DDBJ. So if you're really trying to link your data across three platforms, using the right-hand column accessions is the way to go. And just finally, a quick note on aliases. So you'll see a bit later when we start to do some sample or example submissions that you can define an alias on most objects. We do this mostly to link objects together between your own local system and the ENA accessions. Obviously, you don't get an accession until after you've submitted. So by assigning an alias to an object, you will be able to perform this mapping from your own objects to the ENA accessions, and hopefully this will become a little bit clearer when we do some examples in the following workshop.